Did you know that over 100 million animals are born, poisoned, and abused in US labs every year? Hi, my name is Alan Narvaez and I'm going to talk about animal testing. But first, what is animal testing? The term animal testing refers to the procedures performed of living animals for purposes of research into basic biology and diseases. All procedures, even those classified as mild, have the potential to cause the animal's physical as well as psychological distress and suffering. Often the procedures can cause a great deal of suffering. Most animals are killed at the end of an experiment, but some may be reused in subsequent experiments. Here is a selection of common animal procedures. Forced chemical exposure in toxicity testing, which can include oral force feeding, forced inhalation, skin or injection into the abdomen, muscle, etc. Exposure to drugs, chemicals or infectious disease at levels that cause illness, pain and distress or death. Genetic manipulation, for example, addition or knocking out of one or more genes. Ear notching and tail clipping for identification. Short periods of physical restraint for observation or examination. Prolonged periods of physical restraint. Food and water deprivation. Surgical procedures followed by recovery. Infliction of wounds, burns, and other injuries to study healing. Infliction of pain to study its psychology and treatment. Behavioral experiments designed to cause distress, for example, electric shock or forced swimming. Killing by carbon dioxide asphyxiation, neck breaking, decapitation, or other means. What types of animals are used? Many different types of animals are used around the world, but the most common include mice, fish, rats, rabbits, guinea pigs, hamsters, farm animals, birds, cats, dogs, mini pigs, and non-human primates, for example monkeys and in some countries chimpanzees. But what is wrong with animal testing? For nearly a century, drug and chemical safety assessments have been based on laboratory testing involving rodents, rabbits, dogs, and other animals. Aside from the ethical issues they pose, animal tests are time and resource intensive, restrictive in the number of substances that can be tested, provide little understanding of how chemicals behave in the body, and in many cases do not correctly predict real-world human reactions. Similarly, health scientists are increasingly questioning the relevance of searching aimed at modeling human diseases in the laboratory by artificially creating symptoms in other animal species. Trying to mirror human diseases or toxicity by artificially creating symptoms in mice, dogs, or monkeys has major scientific limitations that cannot be overcome. Very often the symptoms and responses to potential treatments seen in other species are dissimilar to those of human patients. As a consequence, 9 out of every 10 candidate medicines that appear safe and effective in animal studies fail when given on humans. Drug failures and research that never delivers because of irrelevant animal models not only delay medical progress, but also waste resources and risk of health and safety of volunteers in clinical trials. Is there a solution? What are the alternatives? If lack of human relevancy is the fatal flaw of animal models, then a switch to human relevant research tools in the logical solution. The National Research Council in the United States has expressed its vision of a not-so-distant future in which virtually all routine toxicity testing will be conducted in human cells or cell lines, and science leaders around the world have echoed this view. The sequencing of the human genome and better functional genomics, the explosive growth of computer power and computational biology, and high-speed robot automation of cell-based in vitro screening systems, to name a few, has sparked a quite a revolution in biology. Together, these innovations have produced new tools and ways of thinking that can help uncover exactly how chemicals and drugs disrupt normal processes in the human body and level of cells and molecules. From there, scientists can use computers to interpret and integrate this information with data from human and population level studies. The resulting predictions regarding human safety and risks are potentially more relevant to people in the real world than animal tests. This was my presentation. Thank you.